In this lecture, we will do a quick review on cell division, including mitosis and meiosis. But first, here is a prokaryotic cell trying to do some division math. All right, so cell division, what is this? Cells divide by splitting themselves into what we call daughter cells. For cells other than sex cells or germ cells, cells divide to replenish cells that have died. For example, if you get a cut on your skin, cells will have to replenish to fill in the gash on your skin. Non-sex cells or germ cells also divide for growth. As a young kid is growing into adulthood, there is a demand for more cells. And of course, sex cells or germ cells, those divide for the purposes of sexual and asexual reproduction. Depending on the cell type, cells will divide in one of two ways. Mitosis refers to a cell division where the outcome is the exact copies of the parent cells with a full set of parental DNA. This occurs in somatic cells, or cells other than reproductive cells, to replenish growth. Meiosis refers to cell division where only half of the set of parental DNA makes it into the daughter cells. As you may imagine, this type of division is necessary for germ or sex cells. Fun fact, biology is the only science where multiplication and division mean the same thing, where you are referring to cell splitting. Some organisms are asexual and can go through cell division on their own. For example, prokaryotic organisms such as bacteria can divide via a process called binary fission. Here you can see the parental cell, a prokaryotic cell with the prokaryotic chromosomes or the DNA, the plasma membrane, and the cell wall. In this process, the first step involves duplicating the cellular DNA and steering both copies to the opposite ends. So here you have that. The cells then continue to elongate and move away from each other to each opposite end. At last, in step 3, the daughter cells will form as the parent cells divide into two separate organisms. Note that from one parent cell, you achieve two identical daughter cells. Therefore, prokaryotes divide in an exponential manner, base 2. One of the main goals in cell division is to pass on the genetic material from parent to daughter cell. The blueprint of living organisms is in the form of DNA. You may have heard genetic material referred to as chromosomes or chromatin. Since DNA sequences can get quite long, cells came up with a nifty way to organize their blueprint. Chromatin refers to DNA and proteins. It turns out, in reality, DNA is often associated with proteins, and much of their function for these proteins is to structure and tightly organize the DNA. Without any proteins, DNA would be loose and messy, like pasta here. But instead, the cell can organize it to look like a neatly folded telephone cord. Chromosomes refer to an individual pieces of chromatin in their highest organizational order. As mentioned to you in the previous slides, chromosomes are made up of one strand of DNA packed and neatly organized with proteins. Prokaryotes have circular chromosomes, but they do not have a nucleus. Their chromatin can be found in what we know as nucleoids, mitochondria, and chloroplasts. Eukaryotes have linear chromosomes, and their chromatin is found inside the nucleus. You may also find some DNA in mitochondria and chloroplast, but that DNA is circular like that of prokaryotes. Sexual organisms get one set of chromosomes from each parent. Therefore, we have two copies of DNA in each cell, and we call that diploid. Before the cell can divide, the cellular DNA must be copied so that each daughter cell can each get a copy. The copies are called sister chromatids. They are identical to each other, and they are held together by a centromere shown here. This is why chromosomes have an X shape. So what did one sister chromatid say to the other? Stop copying me. The events that start when a cell is formed to when it divides again is called the cell cycle. The cell cycle can be divided into two distinct portions. The first portion is called interphase. Interphase refers to the normal life of a cell, and the cell spends most of its life in this phase, about 90% of its life. The second is the mitotic phase, which refers to the phase of a cell when active division is occurring. Further, the mitotic phase can be divided into two subphases, mitosis and cytokinesis. In mitosis, the nucleus and all its contents like chromatin divide, and during cytokinesis, you get the separation of the two new daughter cells. Interphase can also be divided into three subphases shown here. Remember that cells spend most of their life in interphase, about 90%. In G1 phase or gap1 phase, the cell grows. In S phase, the synthesis phase, the DNA of the cells is divided into two copies, one for each of the daughter cells. And in G2 or gap2 phase, the cell is preparing to divide. After G2, the cell can enter mitosis or meiosis, depending on the type of cell, somatic or germ cell. Now let's discuss asexual reproduction of somatic cells or mitosis. Mitosis is the process where one cell splits into two. The outcome is that the daughter cells must have the same genetic information as the parents. There is also an evenly divide of organelles. The process of mitosis happens in four phases: prophase, anaphase, metaphase, telophase, and cytokinesis, which overlaps with telophase. Together, these events will result in the division of the cell. All right, so as the cells enter the mitotic phase, the cytoskeleton will begin to divide via the microtubules as they form a mitotic spindles that span the cell, spanning from centromeres. So here's the mitotic spindles, and here are the centromeres. You can see the centrosome and the spindles in this cartoon or in the actual fluorescent cell here in green. The microtubules attach to the centromeres to pull and guide the movement of chromosomes to opposite ends. You can see the chromosomes here in blue. During prophase, mitotic spindle forms and the nuclear membrane will start to break. Lastly, the chromosomes start to condense to get ready to split. You can see in this cartoon the early mitotic spindle, the centromere, and the two sister chromatids. You can also see that in late prophase over here, the fragments of the nuclear envelope and the spindle microtubules lining up with the chromosomes. Similarly, in these fluorescent images, you can see the nuclear membrane here, the centrosome here in bright green, the condensed chromatin here in blue, the mitochondria, which was tagged in red, the centrosome, another centrosome over here, and here you have other fluorescent image of the same process. In the next stage, metaphase, the chromosome will line up in the middle of the cell. You can see in this fluorescent image the mitotic fibers, the mitotic spindles right here in green, and the spindle pole, the metaphase chromosomes in blue, and the mitochondria in the outer parts of the cell in red. During anaphase, the sister chromatids separate and are pulled by microtubules to opposite ends of the cell. You can see this in any of these images. Here on the bottom right image, you can see the sister chromatids in green separating to opposite poles. In telophase, the nuclear envelope reforms. Two nuclei and the chromosomes unwind. The final stage of the cell cycle is cytokinesis. Cytokinesis is different between plants and animals. Animals divide using what is called a cleavage furrow, which technically is the construction of the cell membrane that eventually results in the two daughter cells pinching off each other, as shown here. In plants, a mitotic plate is created to divide both daughter cells and create what would become the cell wall. Cells exert very tight control over the cell cycle. This ensures that cells are only divided when necessary. Remember, if you're not actively growing or have a cut or something that needs replenishing of cells, then there is no need to grow. Cells that do not control their cell cycle thus divide uncontrollably. Uncontrolled cell growth can lead to cancer, where our own cells start to divide without any proper control. It's important to understand that while we can treat cancer, we can never really eradicate it from society since it is a malfunction of our own cells. Now let's review sexual reproduction, which refers to as meiosis. This is also a good place to take a break if you need to. Organisms that derive from sexual reproduction are said to have homologous chromosomes. That is to say, two pairs of chromosomes with the same DNA instructions but not identical sequences, each of which is contributed by each parent. For example, in simple terms, we have two genes that code for eye color, one from mom and one from dad. But one of those genes may code for blue eyes and the other gene may code for brown. The gene is the eye color gene, but the instructions are not identical. Autosomes refer to homologous chromosomes that are the same no matter the sex, while sex chromosomes are homologous chromosomes that are different depending on the sex, like X and Y chromosomes for us. We can then differentiate between diploid cells, cells that contain both pairs of homologous chromosomes, like somatic cells, and haploid cells that only contain one set of chromosomes, like germ cells. Haploid cells are formed by meiosis. Here is an overview of meiosis. One cell splits into daughter cells containing half the genetic information. That is one set of homologous chromosomes. Meiosis occurs in two main phases. Each phase is similar to mitosis. So you have meiosis 1 and meiosis 2. In meiosis 1, homologous chromosomes separate. In meiosis 2, sister chromatids separate, thus resulting in 4 daughter cells, which we call gametes. Here is just a written overview of meiosis 1 and 2. Meiosis 1 is most different from mitosis. The end goal here is separation of the homologous chromosomes. No splitting of the centromere occurs here. Notice how in this initial split, the centromere is intact. Meiosis results in 2 daughter cells. The stages similar to mitosis include prophase, metaphase, anaphase, and telophase, accompanied by a 1 referring to meiosis 1. Interestingly, during meiosis 1, an interesting event called crossing over occurs. Specifically, during prophase 1, homologous chromosomes swap arms, dictated here, and make new unique chromosomes. This is important to increase variability in organisms. So here is an overview of meiosis. During prophase 1, here you have the mitotic spindle, the sister chromatids, and the pair of homologous chromosomes. Also, you can see the sites where crossing over is occurring. During metaphase 1, the microtubules attach to each chromosome, and here you can see the centromere of the sister chromatids. During anaphase 1, sister chromatids remain attached, but you can see how they are being pulled to opposite ends. And in telophase and cytokinesis, that one cell is now split into 2 via a cleavage furrow. During meiosis 2, the cell splits once again and goes through these 4 stages: prophase, metaphase, anaphase, and telophase and cytokinesis 2. Notice how during anaphase 2, the sister chromatids separate, which results in haploid daughter cells. That is, they contain half the DNA than the original parent cells did. Okay, so here's a good slide to summarize mitosis and meiosis. In mitosis, somatic cells will duplicate chromosomes. The chromosomes then align and the sister chromatids separate, resulting in 2 identical diploid cells as the parent cell. Does crossing over occur during mitosis? In meiosis, a somatic parent cell will duplicate chromosomes and crossing over will occur. The homologous pairs again align and separate into two haploid cells. During meiosis II, the cells split once again. This time the sister chromatids separate and result in 4 haploid daughter cells where only half of the information from the parent is passed on. So why is sexual reproduction important? Genetic variability is critical for evolution. The more varied a population, the more likely there is an individual that is well suited for the environment. That is to say, as the environment changes, variability allows for organisms to better adapt. In asexual organisms, variability occurs through mutations, such as prokaryotes. In sexual reproduction, variability can increase in one of 3 ways. Independent assortment refers to the genes being split randomly during replication. Random fertilization refers to 2 organisms randomly mating, and we have discussed crossing over between sister chromatids. Okay, so a little bit more in depth about independent assortment of chromosomes. This refers to where homologous chromosomes goes into daughter cells is independent of each chromosome. That is to say, one chromosome from your mother and one chromosome from your father may randomly end up in the same haploid set. This allows for the mixing of parental chromosome in gametes. And also remember that you have crossing over between the sister chromatids. This allows for many possible combinations of the gametes, 2 to the 23rd power, which results in about 8.4 million possible gametes to be produced in humans. Therefore, the reason why you and your sibling could never be identical unless you are maternal twins. That concludes our lecture on mitosis and meiosis. See you next time.